Hello, Chem 213 students, and welcome to our second and last lecture this week about carbohydrates. When we finished off last time, we were talking about the structures of monosaccharides and how by undergoing a hemiacetal reaction, like the ones from chapter 19, having to do with aldehydes and ketones, they can form into a cyclic structure, which is actually the more stable structure. And we discussed some of the reasons why, particularly for glucose. This is an extremely stable structure. As we move forward, we're now going to start to talk about the reactions that carbohydrates undergo, including those reactions that allow them to make long chains that are one of the primary ways that the carbohydrates are packaged when we eat them, and also when they're stored in our body as carbohydrates before they are turned into fats, which occurs when they are in excess in our body. So let's begin to talk about some of these reactions. So number one, the hydroxy groups of these carbohydrates here. These hydroxy groups can react like any other alcohol groups. So they're going to participate in reactions that are that these OH groups, these hydroxyl groups, would generally undergo. These hydroxyl groups can behave as nucleophiles. And so when they do, they can react with, for example, a carbonyl like acetate. And when they do, we can attach an acetyl group here to each of these oxygens. If we use excess acetooxygen here in a pyridine solvent, we can replace all of the hydroxyl groups here. So we have 1, 2, 3, 4, 5 in beta-D glucopyranose. These, in the presence of excess acetoxide here, will result in acetate groups here, acetate esters being formed at each of these hydroxyl locations. No mechanism is provided, but this is simply a nucleophilic attack on the carbonyl group here to produce an ester. It's the same ester synthesis that we've seen in the past. In addition, hydroxyl groups can also be converted to ethers via the Williamson ether synthesis. So recall, the Williamson ether synthesis involves the deprotonation of hydroxyl groups to produce an alkoxy ion. So when we deprotonate these, so deprotonate, I'll just say deprotonate, we get these alkoxy groups here, and there's many of them here. These can then go and do an SN2 attack on the carbon, and the iodine here leaves as a leaving group in a methyl halide such as this. And then we get an attachment of the oxygen here in the deprotonated hydroxyl group to the to the alkyl group here that's on the alkyl halide. So in this case it was a methyl group. So to each of these oxygens on the hydroxy group, a hydrogen is replaced with a methyl group effectively. And silver oxide here is used as a base to deprotonate the hydroxy groups. The most important reactions that these closed chain sugars or these ring monosaccharides undergo is the reaction from hemiacetal to acetal. And so this will occur when we have one ring interacting with another. Just a reminder of what the hemiacetal reaction is if you have an hemiacetal, which was formed by the nucleophilic attack of another group onto a carbonyl group, which turns a carbonyl into a hydroxyl group, and then that nucleophile then is attached to, for example, a alkoxy nucleophile here would be attached to make an ether. So a hemiacetal is a molecule that has an ether group and a hydroxyl group on the same carbon. But this hemiacetal can undergo a further reaction in the presence of acid and another equivalent of the alcohol in order to produce an acetal, in which the hydroxyl group there is ultimately replaced with another ether. And so the acetal has two ether groups attached to the same carbon. So when it comes to monosaccharides or sugars, these can also undergo a similar reaction here with with a neighboring monosaccharide here. And this will result for us in a chain of monosaccharides that ultimately make disaccharides and polysaccharides, which have many, many monosaccharide rings linked in what are going to be referred to as glycosidic linkages. Okay. So here, if we have a ring structure here of this carbohydrate, this monosaccharide, this can react again with an alcohol here in the presence of an acid, and we can produce an ester. However, an ether, not an ester, and this ether can come in two forms here. We can have it on the equatorial position or the axial position, and we'll get a mixture of these isomers here. Or we can have the carbohydrates react with carbons containing hydroxyl groups on another monosaccharide ring. And that's where we're going to get these glycosidic linkages. And so here is the mechanism for the formation of the glycoside. So first we have the protonation of the alcohol, and this should not be too hard to remember, because remember, these hydroxyl groups are terrible leaving groups if they are just hydroxyl groups. They're going to leave as a hydroxide with a negative charge, not a very stabilized negative charge. So the first step in this reaction is going to be the protonation. Of the hydroxyl group that's going to undergo the reaction. So that's what the acid is for. And so we have a proton transfer from the acid over to the hydroxyl group. This gives this oxygen now 3 bonds, 1 to carbon, 2 to hydrogen, and 1 lone pair. That gives it a positive formal charge. And now what we have is a what we'll get here is we'll get this potential for the leaving of the hydroxyl group, and so that would involve this lone pair from the oxygen going down, and a resonance arrow here to form a pi bond, and that leaves this oxygen here with 2 bonds and 3 bonds rather, a double bond here, a single bond here, and 1 lone pair, giving it a positive formal charge. And then the water leaves here as H2O, and so this has resonance too. We have a pi bond between 2 atoms of differing electronegativity, also adjacent to a carbocation. And so if we do this, then it will be this carbon with 3 bonds, 1 to hydrogen, 1 to oxygen, 1 to carbon, and no lone pairs, giving it a positive formal charge. So we get a resonantly stabilized cation as a result of the loss of water from this from this reaction here. At this point, the carbocation is vulnerable to nucleophilic attack. So we can write it in this way, or we can just as easily write it in this way. And in your book, it's starting with this written resonance structure here. But either way, we have a positive formal charge here, attractive to this nucleophile. So that makes it a decent reaction with this weak nucleophile, the alcohol. And so there's a nucleophilic attack on this carbon. We write the resonance arrow back again here to put the lone pair on the oxygen, giving it 0 formal charge once again with 2 bonds and 2 lone pairs. At that point, this carbon here now has 4 bonds, 1 to hydrogen, 1 to oxygen, 1 to carbon, and 1 to this oxygen right here. So it has no formal charge. However, this oxygen, its lone pair was used to make the bond, the new bond, so it has a new bond to carbon and still the remaining bonds that it had before to hydrogen and to this methyl group here. And so it has 3 bonds and 1 lone pair, giving it a positive formal charge. The last step in this glycoside formation here is going to be the deprotonation of this positively formal charged oxygen. And so we have this water as the solvent here, grabbing the proton and leaving it behind an oxygen, and now we have the acetal. Again, this is an acetal because it has a carbon that is attached to 2 oxygens here that are parts of ethers. Now the formation of the beta-glycoside, this involves a nucleophilic attack on the opposite side of the ring. Okay, And so on the opposite side of the ring here, if it attacks, we have the same mechanism here. The lone pair here going on the oxygen, giving it 0 formal charge. The carbon now having 4 bonds, 0 formal charge. This oxygen now having 3 bonds, 1 to carbon, 1 to hydrogen, 1 to this carbon on the ring, and 1 lone pair giving it a positive formal charge. And the final step being the deprotonation of the protonated oxygen here, leaving the acetal with 2 ethers on the same carbon. So you can see that these can both be formed in this glycoside formation, both enantiomers. The one where the methoxy group here is in the equatorial position, and the one where the methoxy group is in the axial position. Now, under strongly basic conditions, aldoses can also undergo what's known as epimerization, and this is where the hydroxyl group on the carbon nearest to the aldehyde in the aldose is inverted in its stereochemistry. And this occurs due to the fact that the oxygen here can be protonated. We have essentially the enol tautomerism. The oxygen here is protonated. We end up with a carbon-carbon double bond here between the two, and this hydrogen atom ends up and this hydrogen atom here ends up transferring over. The one that was on this carbon ends up transferring over here. But now we have this carbon-carbon double bond. This is the ene diol. Ene being an alkene here and a diol. Okay. And then this can then reverse. So this enol tautomerism can go back to having the aldehyde. But when it does, because this carbon is not chiral, the hydroxyl group can end up on either position, and you get a racemic mixture of products. So epimers are diastereomers that differ in the configuration at only one chiral center. So D-glucose and D-mannose here are epimers of one another because only this one hydroxyl group has changed its position in three-dimensional space, indicated in this Fischer projection by being on the left side rather than the right, as it originally was. We can also reduce monosaccharides to alditols, and this reaction is very regularly used to produce sugar alcohols that then go into many sugar-free candies. Now, the sugar alcohols can be partially metabolized to give some calories, but they are mostly not metabolized, so this results in a sweet candy that is significantly reduced in calories due to the presence of these these alditols here, these sugar alcohols, as they're also called. So we can react beta-D-glucopyranose or glucose with sodium borohydride, a reducing agent, and then we can reduce the aldehyde to an alcohol. And now we have a sugar alcohol. Every single carbon has a hydroxyl group attached. Aldehyde oxidation of an aldose produces aldonic acid, and so not only can we reduce the aldehyde, we can also oxidize the aldehyde by reaction with bromine here in water at a relatively lowish pH, lower than neutral. We can oxidize the aldehyde group to a carboxylic acid, essentially putting an oxygen here between the carbon and the hydrogen. A mild oxidizing agent must be used, otherwise the alcohol groups themselves will be oxidized. So we're not going to be using KMNO4 here or chromic acid or sodium dichromate. We're going to use bromine and water as a soft oxidizing agent. So this gives a selective oxidation of the aldose. This doesn't work for ketoses, and it's used as a chemical test to differentiate between the two. So oxidation tests are performed to detect the presence of so-called reducing sugars, which are the Furanose or Furanose forms of a sugar. These include what's known as the Tollens test, reaction with silver nitrate and in a basic environment, and the so-called Fellings test. Glycosides, that is acetals that are not reducing sugars, are not oxidized. So reducing sugar basically means this sugar can be oxidized. And So acetals here cannot be oxidized. So that's the ringed form of the sugar. Only the open chain form of the aldehydes, the aldoses, can be reduced. If we use a stronger oxidizing agent, such as nitric acid, this will oxidize the aldehyde and the primary alcohol of the aldose. So not only will the aldehyde be oxidized here, we can also oxidize the primary alcohol here by the use of a nitric acid. But again, a mild oxidizing agent must be used. If we use a strong oxidizing agent like sodium dichromate or potassium manganate, this will oxidize the hydroxyl groups as well. Now remember, from chapter 19, we also have the so-called cyanohydrin formation, which is a reaction of aldehydes here. So this is simply a reaction of aldehydes with hydrocyanic acid, which will produce for us, essentially we'll have the cyanide nucleophile attacking here, giving us one additional carbon on the cyanide, and the oxygen here that was formerly a carbonyl will then be protonated by the acid as well, making an alcohol. And in this reaction, we're going to get a mixture of enantiomers. We'll get a racemic mixture. And this brings us into the so-called Kilani-Fischer synthesis. And this is in which the cyanohydrin is hydrolyzed to give a carboxylic acid and is subsequently reduced to an aldehyde. So what this allows for us to do is to add an additional carbon atom, right? When we add the cyano group here, we're adding an additional carbon atom. So that's given in blue here. We add this cyano group. Now, if we react the cyano group to produce an aldehyde, we will have an aldehyde with one additional carbon. So if you need to turn an aldehyde into the same aldehyde with one additional carbon, the Kilani-Fischer synthesis is the way to do that. Overall, the Kilani-Fischer synthesis lengthens the carbon chain by one carbon, and as I just said, but retains the terminal aldehyde here. The cyano group can after this reaction here, it can be returned into an aldehyde with one step by reaction with hydrogen in the presence of palladium catalyst and barium sulfate. And so this will produce the C double bond N with the H, ultimately protonating the nitrogen here, and ultimately this leads to, with reaction with water, to an aldehyde. So here's an example of this Kilani-Fischer synthesis where we can turn one, so this is useful for turning one monosaccharide with fewer carbons into a different one with one more carbon. And so we react with HCN. This adds for us another carbon, and so this is the additional carbon right here that's added, and then we finally react with hydrogen and with palladium catalyst and barium sulfate, followed up by water, and the cyano group that we have after the first step is then reduced to an aldehyde. But we have effectively added one more carbon. This carbon here is now this one here. And so we have a carbohydrate with one more carbon. So you can see that it was a aldopentose, 1, 2, 3, 4, 5, diarabinose, and aldopentose. This gets turned into two enantiomeric monosaccharides that are aldohexoses with 1, 2, 3, 4, 5, 6 carbons. But this carbon here that used to be the first one is now the second one. And they are enantiomers with one another, shown by these two groups here being switched. The Wohl degradation does the exact opposite of the Kilani-Fischer synthesis. It will shorten the carbon chain by one carbon. And to do this, we would react with hydroxylamine here in the presence of acetoxide here, and this would ultimately reduce for us the number of carbons by one. The way in which this works is that we produce for us this this compound here with the carbon nitrogen double bond up, and then the nitrogen bonded to oxygen, and then we further oxidize here and turn this into a cyanide group, and finally we have a deprotonation by a methoxy or an alkoxy of some kind to produce a double bond. These two electrons here in the oxygen to hydrogen bond are going to go into making the pi bond between the carbon and the oxygen, while simultaneously the cyano group leaves, and we end up turning an aldo hexose into an aldo pentose by this reaction. Now, the most important acetal-hemiacetal reaction that we're going to study in this chapter is the formation of disaccharides via glycosidic linkage. Disaccharides and polysaccharides. Disaccharides consist of two sugars or two monosaccharides connected in a so-called glycosidic linkage. And glycosidic linkage come in two types. One is the alpha glycosidic linkage. The other is the beta glycosidic linkage. But the glycosidic oxygen here is going to connect carbon 1 on one carbohydrate molecule here, one monosaccharide, to carbon 4 on the other. And this gives us maltose here, a disaccharide. The disaccharide is anomeric here on this carbon and can open. It's a hemiacetal, so it will behave as a reducing sugar. It will open up and produce an aldehyde that can be oxidized. So here is maltose. So notice maltose has what we're calling a 1,4-alpha glycoside. So alpha, because this is going in this direction with this group downward here on the carbon number 1 of the first molecule, and it's connected to carbon number 4 of the second carbohydrate molecule. So there's two ways we're focusing here on glucose monomers, but these glucose monomers can connect with each other in two different ways with the anomeric carbon. So the carbon number 1 is anomeric. When it's in the alpha position and it undergoes the formation of the acetal, remember acetal meaning this carbon has two ether groups on it, one here and one here. If the anomeric carbon has the OH group in the alpha position, this will ultimately produce what's called a 1,4-alpha glycosidic linkage, and this turns out to be the kind of glycosidic linkage that is very, very easily broken down by our digestive enzymes. Whereas, alternatively, if the hydroxyl group is in the beta position when this occurs, you get meaning up and equatorial here instead of down and axial. When this undergoes the synthesis of the acetal, this will produce a beta glycosidic linkage with the hydroxyl group in the beta position here to the 4th carbon on the neighboring ring here, and this beta glycosidic linkage is the one that we cannot, as human beings and many other mammals, cannot digest. Some mammals can, for example cows, which have several stomachs that have bacteria that aid in the digestion and the breaking of this 1,4 beta glycosidic linkage. But for all mammals, we can break this alpha glycosidic linkage. We have digestive enzymes to do that. Only certain mammals and other animals have digestive enzymes that are capable of breaking this beta glycosidic linkage. Lactose is a disaccharide with a beta 1,4 glycosidic linkage. So what this means is that we require a special enzyme in order to break this beta glycosidic linkage. Again, our body has enzymes that are fully capable of breaking alpha glycosidic linkages very easily, but not so many that are capable of breaking beta glycosidic linkage. There is a particular enzyme that is capable of this. It's called lactase, and that enzyme is specifically for breaking the alpha beta glycosidic linkage in lactose. And Some people have more of this lactase enzyme, some have less. People always have it as babies, but often as they go into adulthood, they lose it. In some cultures where historically milk has been drank, there's a greater percentage of the population that has these lactase enzymes and can properly digest lactose. In some, that's of lesser frequency. And those people that don't have that enzyme lactase will be lactose intolerant and will be unable to digest lactose sugar. And so lactose, it's a disaccharide. You saw two disaccharides here, the names, by the way. Two glucose monomers linked in an alpha glycosidic linkage. These are referred to as maltose. A disaccharide here formed from a beta glycosidic linkage is referred to as cellobiose. And the disaccharide formed from a 1,4 beta glycosidic linkage between galactose and glucose form the disaccharide lactose. Sucrose is also a disaccharide. Sucrose is table sugar. Now, sucrose has a 1,4 glycosidic linkage, a 1,4 alpha glycosidic linkage. So this can be easily broken down by our digestive enzymes. Sucrose here, one thing you're going to notice is that it's actually not a 1,4 glycosidic linkage. Sorry if I misspoke here. You can see that on the glucose here, so this is a glucose monomer, it's carbon number 1 that is the acetal in this with the 2 ether groups. But sucrose is special in that it doesn't make a 1,4 glycosidic linkage. It makes a 1,2 glycosidic linkage here, where the first carbon is up here, the second carbon is right here. And the result is that this here is the anomeric carbon for fructose because it's a ketose, and so its anomeric carbon is on carbon number 2 rather than carbon number 1. Glucose is a hexose, so its anomeric carbon is carbon number 1. And in this 1,2 glycosidic linkage, both anomeric carbons are wrapped up in this ether group here. So the rings cannot open up into a straight chain form, and for that reason, sucrose is not a reducing sugar. Its aldehyde groups or ketone groups here cannot be exposed in order to undergo an oxidation, and so sucrose is not a reducing sugar due to the presence of its alpha 1,2 glycosidic linkage rather than a alpha 1,4. And honeybees can convert sucrose into a mixture of sucrose and fructose and glucose. They can basically break this alpha glycosidic linkage as well, the 1,2 alpha 1,2 glycosidic linkage. Now, if we take many glucose monomers and put them in a chain, and they react and form a chain of 1,4 glycosidic linkages, we call that 1,4 beta. If we take a chain of glucose monomers and they make a chain of 1,4 beta glycosidic linkages, like you see here, this is a beta linkage. This will form for us cellulose. Cellulose is also called fiber in our diet. Cellulose is, again, these 1,4 beta glycosidic linkages. We as human beings do not have the enzymes capable of breaking these, so we do not digest this. It passes through our body as fiber, but does help with our digestion, but we cannot get any calories from it. And it will have about 7,000 to 12,000 glucose units linked in glycosidic linkages, and these tend to make up the structural material of plants. And so we're talking tree bark, stems and leaves and so forth. Whereas the starchy parts of the plants, they tend to have a different type of carbohydrate polymer, or polysaccharide as they're called. And that is starch. So starch, like in potatoes or like in the in wheat, not the weedy part, not the shell part, the husk, but the inside kernel part is composed of starches. And starch is also comprised of glucose units, but the glycosidic linkages are alpha 1,4 glycosidic linkages, which we have the enzymes to be able to break. So although we cannot digest cellulose, we can digest starch. And the starch differs. There's some starch that is mostly straight chain here, the some that are branched. So the straight chain one is referred to as amylose, and the branch one is referred to as amylopectin. And so we have a lot of amylopectin in a lot of the starches, and the amylopectin is what gives kind of like like amylose is going to produce crispy textures when it's cooked, but amylopectin will produce the kind of soft and squishier textures. So amylopectin has branches that branch off of the carbon 6 and make a glycosidic linkage between carbon 1 on the new branch and carbon 6 on the place in the amylopectin where it is in the amylose, the long chain, straight chain carbohydrate molecule here. The branches here will form off the 6th carbon with this glycosidic linkage, making for us amylopectin, the branched form of starch. In addition, important carbohydrates, perhaps not as much in humans, but in many sea creatures, for example, like crabs, or many insects like spiders, they have a part of their own structural materials that are formed out of carbohydrates. And this carbohydrate is referred to as chitin. And chitin makes up the shells of spiders and crabs. And these are formed from so-called amino sugars. So these amino sugars, so they're like a polysaccharide here, but on the 2nd carbon we have in chitin, we have an amide group attached instead of a hydroxyl group. And so this acylated glucosamine will form this important polysaccharide, and this is called chitin. And again, this is what composes the shells on many insects and sea creatures. And this one here, this is the carbohydrate monomer here in chitin is like glucose, but instead of having a hydroxyl group on the 2nd carbon, there's an amino group. And so this is called beta-D-glucosamine. Glucosamine, because it's glucose, except for on carbon number 2, instead of having a hydroxyl group, it is an amine group. So they call this the N-acyl group here. So we have an acyl group, meaning we have the carbon double bond oxygen and another carbon attached. This allows for even stronger hydrogen bonding between the neighboring chains, and so what you get is a very strong and tough structure that is ultimately the exoskeleton in insects and arthropods. And N-glycosides they can be formed by a reaction with an amine under acidic conditions. So N-glycosides, what are those? Well, those are what you get if you react a carbohydrate here. This monosaccharide, for example, beta-D-glucopyranose, glucose, basically. When glucose is reacted in the presence of acid with an amine, we end up having that amine make a bond here to this 1st carbon. And this again can occur in the alpha position, the down axial position on the 1st carbon, or in the beta position, the up equatorial position. And this is very important because the nitrogenous base pairs in DNA are going to be connected to the deoxyribose units by these types of N-glycosidic linkages. And these form a nucleoside, which is part of a nucleotide, or the parts of DNA. So here's an example of a nucleoside, which is part of a full nucleotide. The nucleoside here, we have ribonucleosides. So you may know DNA is called deoxyribonucleic acid. RNA is called ribonucleic acid. So a ribonucleoside, like in RNA, ribonucleic acids. Ribonucleic acids are going to have ribose with a N-glycosidic linkage to a nitrogen atom that is a part of a nitrogen heterocycle, or these are what we call the bases in DNA. And so this is an N-glycoside connected at the 1st carbon. Now, in DNA also has these glycosides. However, the carbohydrate here is slightly different from that in RNA. It's called deoxyribonucleic acid, and so it does not have an oxygen atom here on the 2nd carbon. That's the difference between ribose and deoxyribose. So this is ribose, this is deoxyribose. But there's an N-glycosidic linkage here to the base pair, the base in the DNA. So there are 4 bases in DNA. So these are deoxyribose or DNA nucleosides, and they're containing the 4 different nitrogenous bases in DNA. And they're all linked here at the first carbon. And this one here is cytosine, thymine, adenine, and guanine. Notice always, though, it's an attachment to the N at the first carbon. In DNA, the nucleosides are going to be attached to a phosphate group. So we saw that this is a nucleoside here. To get a full nucleotide, you need a phosphate group. So this phosphorus is making 5 bonds with no lone pairs, so it has 0 formal charge. These oxygens here are making 1 bond with 3 lone pairs, so they have a negative formal charge. And here's a whole nucleotide, a DNA nucleotide. And these connect one to another. The phosphate group will connect here on a neighboring nucleotide, and this phosphate will go and connect to a sugar, and then this will go on and on and on and on, and we'll have a DNA strand here formed from this. And so here's an example of the DNA strand. So on the DNA strand, again, this is the first carbon, the second carbon, the third carbon, the fourth carbon, and then the 5th carbon. So at the 5th carbon, that in the deoxyribose, that's what attaches to the oxygen in the phosphate group. And then on the neighboring phosphate group, this is going to attach to the 1, 2, 3, the 3rd carbon, and then the next attachment to the next phosphate is going to be the 5th carbon, and so forth. So for that reason, we have certain namings for the end of a chain like this. At the high end here, or let's look at the low end. The low end here, we have something attached to carbon 3. We call this the 3' end of the DNA strand. And then here at this top part, we have a connection to the phosphate group at the 5th carbon. So we call this the 5' end of the strand. And that's some common nomenclature for DNA strands. The 5' being at the phosphate group here, connected to the 5th carbon in the deoxyribose carbohydrate monosaccharide, and the 3' end being with the connection to the 3rd the 3rd carbon on the sugar. Now, nucleotides, there are 4 nucleotides, cytosine, guanine, adenine, and thymine, and they come in 2 pairs. And if you've taken a biology class and you've learned about this, you probably saw some cartoon like, oh, cytosine is 1 base pair, and this is one of the bases in the pair. And let me redraw this. And you should, in biology textbooks, they often do this. It connects with the guanine, and they connect together, and then that connects to 2 strands of the DNA chain. Now that's a nice cartoon for a simplified version, but what's really going on is when 2 strands of DNA with complementary base pairs are stuck together, they're stuck together by hydrogen bonds. So you can see there's an amine here, a nitrogen bonded to a hydrogen. So this hydrogen is going to be partially positive due to the nitrogen pulling electron density away. This oxygen here is going to be partially negative due to this very polar carbon-oxygen bond, and so we get a hydrogen bonding. The amine here is donating the hydrogen bond. The carbonyl here is accepting it. Then here we have this amine here donating the hydrogen bond, the lone pair on this nitrogen accepting it, and the polar nitrogen here. And then finally we have this amine here donating the hydrogen bond, and this carbonyl group accepting it. And so if you ever wondered what holds 2 strands of DNA together, it is these hydrogen bonding forces. 3 hydrogen bonds between cytosine and guanine bases here. And between adenine and thymine, there are 2 hydrogen bonds, as you can see here. So this is good because when your body wants to replicate the DNA to make a new cell, it can easily unzip these hydrogen bonds, and then when it puts the strands back close together, they will reconnect quite nicely. RNA is structurally different from DNA. First of all, it does not have thymine. It has uridine instead, or uracil rather, as a base pair. And so also the sugar, the carbohydrate, is not deoxyribose, it is ribose. So it has the hydroxyl group here. Deoxyribose does not. So RNA contains uracil instead of thymine, and the sugar is ribose, not deoxyribose. RNA is a single-stranded copy of DNA that leaves the nucleus and goes on to be a recipe, basically an instruction set, to create proteins and enzymes. So an RNA strand looks very similar to a DNA strand, except instead of being double-stranded like a DNA strand would here, DNA would have a second one interacting. It's single-stranded. And the carbohydrate here is ribose, so the hydroxyl group is here. There's not one missing. And we have all the same base pairs except for thymine, which is instead uracil. So in review, we've seen now the structures of carbohydrates and some of the reactions that they undergo. The most important reaction to study for carbohydrates is the acetal and hemiacetal formation. So in this case, we have the hydroxyl group here is going to do, that's a little bit more complex than this. Be sure to go back to section 19.5 if you want to see the full mechanism. But this ring closes when there's a nucleophilic attack on the carbonyl, and we get these rings that are the more stable form of these carbohydrates rather than the straight chain. And we can have these for aldoses. They're also for ketoses. In the case of ketoses, note that the carbonyl is on the second carbon. So in the case of a ketose, you'll see that we've got two carbons hanging off here, whereas in an aldose, we only have the one on the sixth carbon, none on the first carbon. Monosaccharides can also undergo a number of other reactions. So they can be acetylated by the acetoxide and pyridine solvent. They can you can do an ether formation by the use of Ag2O as a base to deprotonate the hydroxyl groups, and then follow that up with an alkyl halide to alkylate that. We can also do a ether synthesis here in the presence of HCl to produce an ether. Right here, we can reduce a carbohydrate from an aldose, for example, to a sugar alcohol, where there's no longer an aldehyde, there's an alcohol on the first carbon. We can also oxidize the carbohydrate from an aldose to a carboxylic acid by the use of bromine and water. And finally, we can oxidize both ends of the carbohydrate, the monosaccharide. So we can oxidize the group on the top to turn it from an alcohol into an aldehyde, and also oxidize the first carbon on the end. So we end up with a dioic acid from reaction of HNO3 and water with heat. Finally, if we'd like to lengthen or shorten the chain of a carbohydrate, if we want to lengthen it, we react with hydrocyanic acid, which will involve a nucleophilic attack of the cyanide ion, and that's going to add one more carbon. That will be this carbon here, and then finally expulsion of the cyanide, and resulting in a racemic mixture. If we'd like to take the the lengthened chain and reduce it, or just reduce the length of a chain, we react with NH2OH here in the presence of the acetoxide and sodium methoxide, which will then reduce this backwards. And so that's a summary of the reactions of carbohydrates, and also their formation of disaccharides and polysaccharides. Disaccharides such as lactose, milk sugar, sucrose, table sugar, maltose, disaccharide from 2 glucose units, cellobiose, also a monosaccharide or disaccharide produced from 2 glucose units, but in the cellobiose, it's got a beta 1,4-glycosidic linkage, but in maltose, it's an alpha-glycosidic linkage. Finally, we have the polysaccharide versions of glucose, very common in food. The starch has the alpha-glycosidic linkage between glucose monomers. The cellulose, which is the structural material of plants, has the beta 1,4-glycosidic linkage. And finally, we ended with this summary of reactions. So I hope that was a helpful lecture, and that will be all for the lectures this week. Good luck on the studying for carbohydrates, and I hope you are fascinated by this new application of the organic chemistry that we have been building up to and applying to biological chemistry. And I look forward to our continued study of biochemical molecules. Have a good one. Bye-bye.